Hello and welcome, it's Dr. Rubisco here again, and today I'm going to be talking to you a bit about the light-independent reaction in photosynthesis. I've already discussed to you where the light-dependent stage takes place, and now I'm going to just go into a little bit of detail into where this light-independent stage takes place. So I'm going to write here light-independent. Stage. Right. So the light-independent stage. So first of all, this takes place in the leaf. So you're probably wondering, well, what are the key components of a leaf then? Right, the top bit is called a waxy cuticle. And the primary function of the waxy cuticle is to waterproof a plant. And it's also to prevent transpiration. Right, so you're probably thinking, well, how does this waxy cuticle actually become waxy? What does it actually use in order to make this wax? But I don't want to go into too much detail about this like in this video, because this is primarily about the light-independent stage of photosynthesis. So this takes place in a layer known as the palisade leaf cells. And the palisade leaf cells are these tightly packed, quite thin, but quite tall palisade leaf cells, which are about here. You have a layer here called your upper epidermis, layer down here called your lower epidermis, and you have another layer here called your spongy mesophyll. Right, so anyway, right, the point of this video is to explain to you about the light-independent stage of photosynthesis. So this takes place in these palisade leaf cells. So basically there's a lot of chloroplast organelles in these palisade leaf cells, and as I've discussed before, I've showed you the anatomy of an actual chloroplast organelle itself with a grainer and all this. I've drawn this multiple times. Anyway, the stroma is just this general space sort of within this double membrane that I've just drawn there. So it's just in general all the space around here. So basically your reduced NADP, which comes from the light-dependent reaction that I've discussed with you in a previous video, is reoxidised. So basically I'm going to draw you out the general cycle, and you'll see sort of where this is reoxidised. First of all, you have your RUBP. Now you won't have come across this RUBP before, but this RUBP stands for ribulose biphosphate, and this combines with carbon dioxide, or CO2, in order to make a 6-carbon compound. But this 6-carbon compound you don't need to know about because it's very unstable, and it quickly breaks down into 2 3-carbon compounds known as glycerate-3 phosphate, that I'm going to abbreviate to GP, okay? So 2 GPs, so I write 2, because there's 2 of them. And basically these GPs are then formed into a molecule known as triose phosphate, otherwise known as TP. And then RUBP is reformed in a phase here called the regeneration, just because it's the regeneration of ribulose biphosphate. Okay, so we know this. So where does this reduced NADP come in? Well, basically in this process between between the anabolic reaction in which RUBP joins with carbon dioxide, this bit here is where this NADP is reoxidised, so reduced NADP from the light-dependent reaction forms NADP again, which can be used again in the light-dependent reaction. So if you remember that it's reduced, then supposing it's reoxidised and it loses that hydrogen, it can gain it again and be recycled essentially. So now we've got a GP. Oh yeah, I forgot to mention also there's some ATP breakdown here, or ATP lysis, or whatever you want to call it. ADP and inorganic phosphates formed here, and the same thing happens over here. So you've got ATP going in and ADP and inorganic phosphate going out. Right, so now I've cleared that up. the next thing is to know what this GP is used for. Well, glycerate phosphate, I'll write it up here. GP is used in order to produce fatty acids, but it's also used to produce amino acids as well that can be built up into proteins and stuff. So, amino acids and fatty acids. Right, so fatty acids, what they can do is they can combine with something called glycerol, which TP can be used to form. So right down here, TP can form glycerol. Glycerol combines with fatty acids in order to form lipids, which can be triglycerides, which means that they've got one glycerol molecule for every 3 fatty acid chains, hence the term triglycerides, because tri means 3. Okay, so that's essentially what GP and TP can be used for, although TP isn't only used to make glycerol, it's got many other uses also. So it can, so because this is a free carbon molecule, it takes 2 of these to combine to form a hexose sugar, so to form like glucose, and then I'll write up here. So if you have glucose, and it can form fructose as well, because glucose can be converted to fructose via an enzyme known as isomerase. So once glucose is converted into fructose, like some of the glucose still remains as glucose, and then combines with the fructose in order to form a sugar that's known as sucrose. I don't know if you heard of sucrose, but basically that's a sugar that's used in plants for the process of translocation, which basically means moving, in order to move like water and like other substances up the stem, you need to use this sucrose. It's basically just essentially used in transport. So glucose and fructose. You also got things like cellulose that's formed, and cellulose is used in plant structure. It's actually used in the plant cell wall in order to provide it its sort of structural role. Starch as well can be built up from this, and that's pretty much it. Okay. So the one thing I mentioned is that there's also another enzyme that's an anabolic enzyme that actually joins this RUBP to CO2, and this is called Rubisco. Now, as you probably guessed, this is biology, so it's not going to have an easy name to remember like Rubisco. It's actually called ribulose biphosphate, which is the same as the RUBP bit, but this isco bit at the end just means that it has it carboxylates essentially, so it's called ribulose, which means it adds the carbon dioxide to the RUBP, so it's ribulose biphosphate. Otherwise, it can also be called bisphosphate, but you don't need to worry too much about that. Ribulose biphosphate, and then the last bit is carboxylase, because it adds the carbon dioxide to the RUBP, or ribulose biphosphate. So that is your process of light-independent reaction. If you have any questions, feel free to leave them in the comment section of this video. I'll see you in the next video. Thanks for watching.